Okay. Yes. So yeah. So there are some problems with the network and other things. Divyanshi should be able to join again. She got removed, I guess. Okay. So we were talking about in the previous one, we have discussed about their general scheme. We were also able to discuss about the approach they have taken, um, and several other factors, right? They use the Yamanaka factors, then they have used the self-cleaving peptides like these, and then they were able to generate in a monocistronic mRNA, they were able to generate 4 different proteins. That is the answer for one of the questions in CAA 2. Okay, and then we also discussed about at the level of protein, they have shown that they are produced in an adequate amount. And then we also discussed about the selection procedure, what it means if it is able to grow on neomycin, and if it is unable to grow on neomycin, what it means. Positive and negative selection, that is what we have seen. In here, the selection is used to distinguish between if the piggyback transposon is excised out and it is whether it is reintegrated or not. What we want is excision should have happened, but reintegration should not have happened. This is what we will need. In that case, they would be puromycin sensitive and FIAU resistant. Puromycin, that selection is for positive selection, and FIAU is negative selection. So then we also discussed about RT-PCR and southern blotting and so on. And then about sequencing, they have proved that upon deletion upon excision of piggyback transposon, they have reverted back to the sequence that was similar to that of fibroblasts. So we have also discussed about RT-PCR analysis and also about the bisulfite sequencing. So we just have 2 slides. So we will rather go slow, and then we will stop, and then I will probably have a general discussion. So this is the construct they have made, as is shown in here: ampicillin, piggyback vector, the promoters, the OCT4, SOX2, KLF4, and CMYC, and the polyadenylation signal. So the green, the ones included here are the probe sequences. Okay, so they use that particular probe to identify each of these genes, whether they are present or not, and so on. If I should remind you about what is 25. That is, in the previous ones, IPS 25 they call. That is, so they have the differentiated cell, they have transfected with piggyback vectors, with OKSML, say, and then they have got IPS. So they must have gotten more than one IPS cell, right? They would get, and there some of them they have labeled as, one of it they have labeled as IPS 25, another one they have labeled as IPS 28, and so on. IPS 216 also. And these are 3 different cell lines you can assume that they have gotten, which are induced pluripotent stem cells, and from each of them they have now derived they have removed the piggyback vector. Okay, excision has happened, because of which they would call, they can get more, right? 1, 2, say here. So that they would call as IPS delta, IPS 25 delta 1, or IPS 25 delta 2, like that. That means this 25 indicates the cell, induced pluripotent cell with the piggyback vector in it. Delta 1 and delta 2 are the cell lines in which the piggyback transposon has been excised. Similarly, for 28 and delta 6 and delta 8, 216, 13, and 12 and 13. Okay. So they have done the analysis with southern analysis, and they have used OCT4 probe. So you can see that here is embryonic stem cell did not have this additional band. Why? This is a natural normal one. Okay. In 25, they have integrated this whole vector, this piggyback transposon. They have integrated, sorry, from here till here. Okay. Oh, wait, let me get it right. So this whole part has been incorporated in some place. Okay. That is why there is an additional band you see here. This band you do not see here, but you see here. What it means is that particular band is present is because of finding this OCT4 gene. Okay. So you can see that the normal embryonic stem cells do not have it, but the IPS 25 has, and IPS 25 delta 1 and delta 2 do not have. What does that mean? It means that excision has happened. And you do not see any additional bands in any of these cases, which also confirms that reintegration of the excised piggyback vector did not happen. Okay, that is the same way they have shown for each of the things in each of these case cell lines that they have gotten, and for each of the genes as well: OCT4, SOX2, KLF4, and CMYC. Okay, in here, this is where you should compare. So when you are looking at gel pictures, it's actually a good choice, a good way of reasoning or deduction. The normal cell line embryonic stem cell does not have it, but the 25 has it. That has got incorporated. That is the integrated one. And then delta 1 and delta 2 do not have it in this place, which means that that is the band that is the transposon, piggyback transposon is excised. Okay. That is the way they have shown for in all the cases. So when you look at the gel, you should know about the nature of the controls. What is the control used? I mean, control is will add relativity. In the sense, we are comparing our test samples against in relation to the control. There is a standard for comparison, and then you can figure out what is different in the test in relation to the control, and then you can deduce your conclusions from it. Okay. So now we have to focus on this particular one. I think I have described about chimera. What is a chimera? And we will see about chimera today. But the question about why they are why is it included in this part is they had differentiated cells. They made induced pluripotent stem cells. They have verified that the sequence is fine. They have verified that there is no reintegration. They have shown in every way that is something comes under quality control. One has to ensure that the induced pluripotent stem cells that were made do not have any other genetic modification. They should be equivalent to that of any embryonic stem cells isolated. And once that is assured that they are induced pluripotent stem cells, and that there is no integration or any other kinds of genetic manipulation, now we have to prove something else. All embryonic stem cells are pluripotent. What is pluripotency? Pluripotency is the ability to give rise to all cell types. When you say all cell types, predominantly we have ectodermal cell types, mesodermal, and endodermal um, cell types, tissue cells arising from these three primordial germ layers. That will give rise to all the cell types in our body. The difference between totipotency and pluripotency is, in totipotency, the cells would have capacity to give rise to a complete organism, whereas pluripotent one can give rise to all cell types but not as a complete organism. Okay, so we have to prove now that the induced pluripotent stem cells are actually pluripotent and they can give rise to all cell types. If we have to prove this, then we have to use manipulation technologies involving manipulation of embryos. I think in when we were discussing about transgenic animals and we were discussing about embryonic stem cell technology, where engineered embryonic stem cell technology, where we take these stem cells, we make the genetic modification, and then put it into an embryo, into a blastocyst. So I hope you know where embryonic stem cells normally come from. Embryonic stem cells, I will probably use the next slide. Embryonic stem cells come from blastocyst. So a zygote will give rise to a morula, that is all a bunch of cells all looking the same. Differentiation has not occurred. And then we have the blastocyst forming. It is the first time the cells would have differentiated. They will differentiate into tophoectoderm and into inner cell mass. The green ones are the inner cell mass. Inner cell mass is the source for embryonic stem cells. Okay, normally anytime somebody says we isolated embryonic stem cells, that means you destroyed an embryo, and they would have isolated the ICM, which are cultured and called as embryonic stem cells. The embryonic stem cells have the pluripotency, meaning that all the cells in our body, all types of cells we have in our body, are derived from this ICM. Okay, because if a patient is there and the patient needs some tissues and you want to have his or hers pluripotent stem cells, you cannot get the embryo of this person. The person is already grown up. So what we should then do is we should take his differentiated cells and then make them into induced pluripotent stem cells. The embryonic stem cells here and induced pluripotent stem cells are almost same. The major difference is where do they come from. Embryonic stem cells come from inner cell mass from a normal embryo, blastocyst. Whereas induced pluripotent stem cells are usually obtained from the person's differentiated cells, and we are inducing it by artificial means. That's why we call it induced, and we are making pluripotent stem cells out of it. We are genetically reprogramming a differentiated cell into pluripotent stem cell. Because we can then get any type of cell, whichever cell we want, and then be able to put it into the patient when required. So before we do that, we now have to prove that the stem cells that we made, or the pluripotent stem cell that we made, are actually pluripotent, and then they can contribute to the potential that we are thinking, that they can give rise to all cell types. So what we can do is that we should first take a blastocyst. So assume that, like what we are doing in our experiment in the previous one, we have now got induced pluripotent stem cells. Now we need to figure out if these induced pluripotent stem cells are really functional or not. So we will take them to be something like this. Okay. So what we should then do is take a blastocyst. We will take a blastocyst. Okay, assume that you should all remember that all the experiments that we are doing are not done in humans. It is not permitted in most majority of the countries. Okay, if it is happening, mostly it could be illegal, or maybe some countries have given permission. I think China and Korea, they may have had temporarily, they may have given permission to work on human embryos, but largely they remain in research, or maybe highly restricted or regulated. So there is the study that we are also studying, looking at, is done in mice or so. So keep that in mind before you assume anything else. So we are taking a blastocyst, a normal blastocyst, and we will introduce the induced pluripotent stem cells into this blastocyst. Okay. Now, what will happen if I go back to a little bit on this case? If we allow this whole embryo to grow, and then it will eventually give rise to a mouse. Because I'm taking green color, so the whole mouse would be in green color like this. Okay, just for sake of understanding. Now, if here you can think of in the second case, in this case here, you have two different types of ICM. One is the original green one that has come from the donor, the blastocyst that we have chosen, and the other one that we have made, induced pluripotent stem cells. And if this cell, if this embryo has grown into a complete organism, it would give rise to a chimera. What is a chimera? A chimera is an organism that has two different cell sets. I will describe once again. So all the green parts of the body must have been generated from the green ICM. All the red parts of the body must have been generated from the red induced pluripotent stem cells. And this is a chimera. You can have multiple definitions. In all the cases, you can think of it as cells derived from two different ICM. If it's an organism derived from two different ICM. So you can also have very rarely developmental issues. I think I must have described this somewhere, but if not, I will try to do it again. I hope you have seen conjoined twins, right? Conjoined twins. They get, say, the two humans fused with bodies are fused together, and they have to lead life together, conjoined all the time. Sometimes the head is fused, sometimes the body parts are fused. And if we try to separate, not all can be separated as long as there are no shared common essential organs. Then probably you can try to separate them, but it's not the case in majority of the cases. Either the bodies or the heads are usually fused up. So how do they arise? And in some cases, there is also a situation where one may have a body, and some parts of the body are actually made, the conjoined twins become a part of the original body. So morphologically, if you look at this person, they will look as an individual, single individual. But there is one underdeveloped embryo or underdeveloped body parts within this person. Okay. How do they arise is what I'm trying to now describe. They arise because I hope, I would also want to discuss a little bit about twins. First of all, identical twins, how do they form? Identical twins form something like this. There is one embryo, okay, one zygote, it is dividing during modular stage or so. They might separate into two, and each grows into an individual, and their genome set is exactly same, and because of which they will look identical, right? Non-identical ones, they are from two different um, what do you call that? They are from two different embryos, in the sense there are two egg cells, and they are each fertilized independently by two different sperms. And so this one is going to grow into one individual, and the other one is going to grow into another individual. They don't look, they're born at a time, but they do not look similar. So the conjoined twins I was talking about, sometimes what may happen is they may be, um, you may have assume that there are two different, like I was describing, two different fertilizations have happened like this. But during somewhere around this, assume that they have, I'm trying to, yes. So if these two fuse up, so now this is one embryo and this is another embryo during modular stage or something, assume that they have fused up. Okay, I'll just take one as red and one as blue. And over the period of time, because of something, they might have fused up into one. Okay, and this might give rise to one individual. Something like this. So what do you make out of it? This person is a chimera. This person is growing, has body cells that are actually that are derived from two independent fertilization events. This is one fertilization, fertilization 1, and this is fertilization 2. This is one embryo 1, and this is embryo 2. And they've both fused, and they have produced one single individual, which is having some body parts made from one embryo and some parts made from another embryo. That is a chimera. The same technology can be used to determine if the produced induced pluripotent stem cells can give rise to cell types or not, all cell types or not. We don't go and check all there are about 210 different cell types in our body. Nobody's going to go and check all of them. That will take a lot of resources, time, and efforts. So what they would normally do, I hope you understood chimera, then we can go and see the results there. So when they did the experiment, what they are looking for is something like this. I will try to explain the genetic construct a little bit, and then we will try to see what we get. So they have taken the title of the figure, contribution of integration-free iPSCs to somatic tissue and germline in chimeras. Okay, DPC is days post-coitum. The chimeric embryos. So you have, like I described in the the previous one, they have taken the so one of it is non-chimeric embryo. The one on the right side is the non-chimeric, and sorry, I'm talking about, this is AB. Sorry, I'm talking about the B. The top one is non-chimeric, and the second one is chimeric one. Okay, in this one, it is both the same. A, you have to compare figure A with this one. One second, let me erase these. This is postnatal day 5 pups. B is derived from integration free with constitutive GFP. Which one is GFP? The green one is, of course, the GFP. Should they say? Okay, we will just go with the second one, and B will come back to it. Non-chimeric is A. In the non-chimeric embryo, and pup embryo and pup. Okay, well, I'm sorry now. It's so what they have done is at different stages. Okay, first one is at the level of embryo. Second is at the level of pups. And third, they're looking at is for the development of gonads, germline, isn't it? So they are tracing it, what happens during embryo, how is the contribution during embryo, how is it during pups and gonads. In A, we'll just see the embryos. This is the normal, this is the one with induced pluripotent stem cells. You can see that the whole body is giving the fluorescence, which would mean that the induced pluripotent stem cells are contributing to various types of body parts. Okay, this would be the control in that case. If you see in B, at the pup stage, the top one is normal one, and the second one is the one with the induced pluripotent stem cells. That is the chimeric one. So you can see again that it is showing all the it is showing the close shot even at the level of pups, especially in the brain and other parts. It is showing large amount of contribution from the induced pluripotent stem cells. The third is in the last is at the level of gonads. Of course, I think you know that one can mix the gametes of donkey and a horse and get a mule, right? Does that mean these two can interbreed? No. Why is that not so? Because all mules are sterile. That means the genes, the formation of babies is not enough. These babies should be able to transmit the genes and produce more organisms. So we should say viable and fertile babies, progeny. You can say how you define species. Species is defined as those that can only breed within themselves, among themselves, and not outside, to produce progeny, fertile progeny. So here we are putting induced pluripotent stem cells, and we should ensure we know from the figure A and figure B, we know that it is contributing to all cell types. But if it is not contributing to the germline, then it is a problem. So that is where in C, they're showing that the gonads are all the induced pluripotent stem cells are contributing to the gonads as well. Why are they in green color? They should have made some genetic modification again to show that. So in that case, typically it would be something like this. They would have put EGFP, E for eukaryote, green fluorescent protein, and they should have put a constitutive promoter. And this is GFP. So this any cell having they should have made genetic manipulation in the iPSCs that are generated. In the iPSCs, they have integrated this the EGFP for reporting. Understand that this is not for clinical trial or anything. This particular experiment is to prove that the induced pluripotent stem cells that were made are have the ability to contribute to all types of tissues, including the germplasm. So they would have to prove that they need a reporter marker to distinguish between the induced pluripotent stem cells from the normal ICM of the donor or the foster embryo. So they must have made a gene construct like this and put it into iPSCs, and these iPSCs were put into an embryo and made a chimeric embryo is made from which the organism is developed, and that organism shows different parts illuminated, something like in figure A and B and also C. In this case, they must have used constitutive promoter. What will happen if they used tissue-specific promoter? Assume that if they have used only a promoter that is specific to, say, neurons, then only the brain and the spinal cord might actually glow, and nothing others may not glow, right? So they want to prove that they can form any type of cells, so they must have used a general constitutive promoter, so that GFP is expressed anywhere in all of the iPSCs. Even though they have differentiated, they still should be able to express eGFP, so that we can observe and confirm if the cells iPSCs are made have the ability for pluripotency or not. Okay. Yes, that's the end of the chapter. And any questions, by the way? Have you got the chance to read this chapter yet or no? This particular paper? I have sent the paper. I encourage you all to read the paper as soon as possible so that you can be in touch with what I have discussed. And if there are any questions, you're more than welcome to ask. Okay. Anybody has any questions?